Bismillah Rahman Rahim. Welcome back, friends. Welcome to another video lecture by Kami Biology. Today in this video lecture, we are going to talk about the phagocytosis in a detail. Okay. Now, first of all, what is phagocytosis? Phagocytosis is the phenomena in which the microorganisms are engulfed by the immunocells. Now, what kind of the immunocells they are engulfed means WBC. So WBC have the ability, they are the immunocells, they can engulf the microorganism. So in this first video lecture, I will just explain the summary of the phagocytosis, that what actually happened in the phagocytosis, and then in the step-by-step video lecture, we will explain the phagocytosis in much more detail. So let's explain. Phago. So first of all, let's explain this term. As the name suggests, phago. Now, phago is actually derived from the Greek word means phagenes, which means what? Which means to eat. To eat. And this cytosis means cytone. Cyte. It is actually, what does it mean? It means cells. So those cells which can eat or which engulf the microorganisms, bacteria or pathogen or antigen, or we can say pathogenic particles. Okay, so we call that phagocytosis. We call these cells phagocyte, and the process are known as phagocytosis. Okay, now if I explain it, one thing more about the phagocytosis: when the bacteria are encoded with an antibody and when the phagocyte engulf it, then we call that opsonization dependent pathway. If the bacteria don't have, or we can say if the bacteria doesn't encoded by the antibody, and when our phagocyte engulf it, so then we call that independent opsonization pathway. Okay, so what actually happened, if I explain it with the help of diagram. So first of all, what actually happened, let's suppose, we have different kinds of cells, means we have macrophages, dendritic cells, neutrophils, monocytes. So all these cells are actually what? All these cells are the phagocytes. So let's draw a simple diagram of the phagocytes. Let's say this is the macrophages, or let's say dendritic cell, or any kind of the phagocyte. So let me draw a cell. No, let's suppose this is a cell. Let's say this is a macrophages. First thing, these cells have different kinds of receptors. Okay, these cells have MHC1, MHC2. These cells have LPS receptor, toll-like receptor, mannose receptor. So let me draw each of the receptors. So they have different kinds of the receptor. They have MHC1 receptor. Now let's suppose this red one representing the MHC1 receptor. They have MHC2 receptors. So one thing, MHC2 receptor are not usually present. It may be formed when the bacteria are converted, or when the microorganisms are converted into small fragment, then this MHC2 molecule or MHC2 receptor will be formed. They have also another receptors which we call it LPS receptor, lipopolysaccharide receptors. So this is another receptor which we call it LPS or lipopolysaccharide receptor. Moreover, they have some special kinds of receptors which have the unique features. We simply call that what? We simply call that toll-like receptors. So let's suppose this is the toll-like receptors. All these receptors, as you are watching, all these receptors are called what? All these receptors are called pathogen recognition receptor. So all these are called what? Pathogen recognition receptors, or we call it pattern recognition receptors. Now what actually happened? Let's suppose there is a bacterial species as well. So let me draw a bacterial species here. Let's say these are the bacteria or euglena or fungi, any kind of microorganisms. Let's suppose these are. Now what actually happened? Sometimes phagocytosis phenomena happen. If these cells have the ability, let's suppose these are what? These are the phagocytes. Let me write. Phagocytes. Now let's suppose if this cell have the ability to engulf this microorganism, then there is no chemotaxis factor, or there is no chemokines are released. But sometimes these cells don't have the ability to engulf this bacteria, because sometimes this bacteria have capsule, or sometimes they don't have the capsule, but still this phagocytes cannot engulf it. Now, after that, what actually happen? They will secrete some kinds of the signals which we call that chemokines. But let's say this immunocells or this phagocytes have the ability to engulf this microorganisms. Then they don't produce these kinds of the chemokines. Then what actually happen? Such bacteria have a special kinds of surface molecules on their surface. Each bacteria, each cells or each microorganism have special kinds of receptor or special kinds of surface molecule on their outer surface, which we usually call that pathogen associated molecular patterns or PIMS. PIMS means we have, on the bacterial cells, we have lipopolysaccharide, we have mannose, we have some other receptors also. We'll be talking in the next video lecture. So due to this way, these phagocytes have the ability to engulf it. Now, after that, once these bacteria are engulfed, so then what actually happen? After that, this cells, these bacterial cells are now engulfed. So let's suppose this shape represent that each of the cells are now engulfed. So once it is engulfed, and once it is engulfed, then a phagosomes is formed, or we call that vesicle. A vesicle is formed. So what actually happened? A vesicle is formed. So inside each vesicles there are what? There are the bacterial species. Now after that, what actually happened? This, phagosomes are fused with the lysosomes and it make a complex which we call that phagolysosome. So let me draw. So let's say this is again a phagocytes. Now this one represents what? This one represents the lysosomes. And this is what? This is, let's suppose, the vesicle, or we can say phagosomes. So they make a complex which we call it phagolysosomes. Phagolysosome. Now once the complex are formed, so then what actually happen? The bacteria which are present at that area, so then these lysosomes have different kinds of hydrolytic enzymes. We'll be talking in some other video lecture, which we call it hydrolytic enzymes, lysozymes, H2O2, hydrogen peroxide. Okay. Now after that, what actually happened? This phagolysosomes once formed, so then different kinds of the chemicals are released. So what actually happened? Chemicals are released. Now after that, what actually happened? With the release of each chemicals from the lysosomes, which we call the hydrolytic enzyme H2O2 or lysozymes. Due to this way, this bacteria will be break down into small fragments. So after that, what actually happened? The bacteria will be break down into small fragments. And this is the bacteria which is break down into small fragment, or any microorganisms. So it is break down into small fragments with each antimicrobial chemicals. So let's suppose this complex contain what? Antimicrobial chemicals. Antimicrobial chemicals. So then once it is converted into small fragment, then what actually happen? After that, MHC2 molecule are formed, or we can say that these molecules are now represented to some other amino cells. So after that, let me draw another cell. So then it is represented to MHC2 molecule. Now this is the MHC2 molecule. So on the MHC2 molecule, these are what? These are the antigenic fragment or peptides. Now each peptide is then represented to some other cells. Means helper T cells, lymphocytes, T lymphocyte, B lymphocyte, different kinds of the cells. Each fragments are represented. After that, what actually? Sometime each fragments are represented to the immunocell, or sometime these fragments are fused to this, or we can say this phagolysosomes complex are reached to the cell membrane area, and then they burst means this lysosomes or this complex are burst, and they will release each debris. So each debris are then released out. Sometimes each debris are what released out to the from this cell. Okay, so sometimes this phenomena happen, sometimes this one phenomena happen. Now, so that's all that's all are the actually the summary of the phagocytosis. Now here are the questions are usually arise in our mind, that this bacteria are these bacteria are engulfed by our phagocytes, means our own cells, but our own cells cannot engulf the host cell, means our own cells, or we can say that each microbes we can say that each phagocytes doesn't activate it against our own cell, but it is activated on the bacterial cell. What are the reason behind that? It is due to this PIMS. Okay, it is due to this PIMS. So due to this PIMS, the bacteria are engulfed, but our own cells are not engulfed by these phagocytes. So inshallah in the next video lecture I will explain this PIMS in a detail. Okay, so I hope you understand about this phagocytosis process. If there is any questions in your mind, so you can share it through comment. Inshallah I will give you answer as soon as possible. If you like this video, so make sure to hit the like button, share this video, and also subscribe the channel, and stay tuned because in the next video lecture we are going to talk about this PIMS in a detail. Thank you so much for watching.